So following on from the previous session where we were looking at very general analyses like regression, correlation and analysis of variance and the ways to visualize those analyses, in this session we're going to be focusing on 4 very specific types of analyses. So we'll look at how to analyze size class distributions, how to quantify interspecific aggregation and interspecific associations, and how to identify vegetation types using multivariate ordination. Let's first recap a little bit on the previous session. So what do you think would be an appropriate analysis to test the relationship between soil depth and species richness? Take a second to think about what analysis you think would be appropriate and how you'd visualize that data. Well, because both those variables are continuous variables and because we expect soil depth to influence species richness, not the other way around, I think we could say that regression would be the right analysis tool, and we could visualize the data as a scatter plot with species richness on the y-axis and soil depth on the x-axis. A second question, what analysis do you think would be appropriate to test the relationship between topography and species richness? Now remember, of course, that at Magaliesberg site we measured topography as a categorical variable. It would either be a convex, concave or flat topography within the 4x4 meter plot. So what analysis do you think would be appropriate? Well, here I think analysis of variance would be appropriate because our predictor variable, topography, is split into 3 categories, where species richness, our response, is a continuous variable. Let's make this a little harder now. This isn't something that we've covered. But what analysis do you think would be appropriate to test how soil depth and topography together affect species richness? See if you can come up with a way that we can analyze those 2 predictors simultaneously. Well, one option here would be to split the data set into 3 different data sets representing the concave, the convex and the flat topographies. And then for each of those data sets, you could plot the relationship between species richness and soil depth. And what's interesting here is that we see contrasting patterns in the effect of soil depth on species richness in the 3 topographies, although none of those are statistically significant. Okay, just to bring back the slide where we were talking previously about why we were collecting such a wide range of data, and the reason for that is so that we'd be able to answer a diversity of different questions. And that's just highlighting those questions include understanding size class distribution, documenting species dispersion. Okay, both of those are population characteristics. We could test for spatial association between species, which is looking at interactions just between pairs of species. And then finally we can look at identifying plant communities, which is taking a community approach. Okay, so we're going to start by looking at how to document and how to interpret the size class distribution of species. Essentially the tool for this is just a histogram, which is a graph that shows the abundance or proportion of individuals of different size classes. So here, for example, we have the histogram for Aloe-Great-Headii and then for Euphorbia shinzii, so both common succulents at the Magaliesberg site. And what you can see here is that there's slightly different shapes for the 2 of them. So for Aloe-Great-Headii we see that circumference follows an approximately hump-shaped pattern, where most individuals are found in these middle size categories, with few smaller individuals and a few larger individuals. Euphorbia shinzii, on the other hand, shows a declining pattern, where generally the larger individuals get, the less frequent they are. Now, how do we interpret this? What does this mean biologically? Well, if we assume that size is correlated with age, and that's an assumption that is sometimes valid, sometimes not, but let's assume that size is correlated with age for the species we're considering, then the size class distribution tells us about the recruitment and the survival of individuals within that species. So again, let's take two species from the Johannesburg, the tree Ochna pulchra and Xerophyta retinervis, the Bobbejaanstaart, the baboon's tail. What we can see here is that for Ochna pulchra we've got that approximately consistent decline in the frequency as we go to bigger plants. And what this suggests is that there's what we would call continuous recruitment. Every year new seedlings establish, and every year some of those grow into the next size class, but the majority die, or a large proportion die. And that continues year after year after year, and we end up with only a few large individuals surviving. We see a very different pattern for Xerophyta here, where there's no evidence of a trend for fewer larger individuals or more larger individuals. And this suggests that we're seeing sporadic recruitment, that the number of seedlings entering a population or entering a specific size class varies from year to year. So by looking at these size class distributions, we can make a guess about the recruitment pattern for these species. Okay, if you want to run these analyses for your Johannesburg data, if you're working in Excel, you can just go to the data analysis tool and click on histogram, whereas in R you would just use the hist function and put in the variable that you want to draw the histogram for. Okay, so moving on to the other population characteristic we might want to document, and that's the spatial pattern that species shows. This can also be called its dispersion, its clumpedness, or its local distribution. Now the three panels in the middle show uniform, random, and aggregated or clumped distributions. Clumped is the easiest one to understand. We can see that those points, which are representing individuals of a species, are clumped or clustered. Uniform distribution, on the other hand, is where there's an approximately equal spacing between all individuals. And then in between we've got the random distribution, where there's a mixture of large and small distances between individuals. Now there are various ways that one could quantify this, and one of the simplest and oldest ways is an index of dispersion, which is also more descriptively called the variance to mean ratio. And this is a good measure of the degree of randomness in any given phenomenon, and we can use it to describe the randomness of the association of plants of different species. So this is a nice way to kind of represent it, I think. Let's start in the leftmost figure. There you can see we've got 4 quadrats where the species is abundant, and the other 12 quadrats the species is absent. So what do we see here? Well, this is obviously a clumped distribution. Because there are only 4 quadrats where the species is present and 12 where it's absent, we see a very high variance in this data. Variance is representing the variability in the abundance of the species in the different quadrats. And because this variance is so high, we see a really large ratio, a ratio of 125 to an average of 6.25. So we have a ratio much bigger than 1, indicating a clumped dispersion. Moving on, for a uniform dispersion, there's very low variance between the quadrats, right? There's approximately the same number of individuals in each quadrant. And because variance is low here, the variance to mean ratio will be less than 1, indicating a uniform distribution. And then in between the two of them, the random dispersion, we see that the ratio of the variance to the mean is approximately equal. Okay, so this is great. We can easily quantify the dispersion index. So we've got a metric which tells us about interspecific dispersion or clumpedness. But how do we interpret this ecologically? Well, by looking at this pattern, we can suggest what processes, ecological processes, are occurring in the system. So this can allow us to make inferences or suggestions about dispersal, survival, or even interaction patterns within the species. The problem here, though, is that many processes can give rise to the same pattern. So this isn't hard evidence, it's just a suggestion of what's driving the system. Now, let's try see if we can understand this by looking at 4 examples. So, what type of dispersion or local distribution would you expect for a species that is dispersed through endozoochory? Remember, endozoochory is when animals transport seeds internally. I think here we would expect a clump distribution for a species if that's how its seeds are distributed. Of course, the reason for this would be that every time a bird defecates or regurgitates seeds, there's going to be a cluster of seeds in one spot, and therefore we expect there's going to be an aggregated distribution of individuals. Second example would be, what type of dispersion would we expect where seedlings are heavily impacted by diseases from mature plants? In this case, we'd expect a uniform distribution, and the reason for that is that we're seeing this negative interaction, this negative effect of mature plants that can easily act as a source for diseases which affect the seedlings. As a result, we expect seedlings to occur far from host plants so that they're safe from the diseases carried by the mature individuals. Third example, what type of dispersion would we expect for species with very strong intraspecific competition? Now, of course, one of the consequences of strong intraspecific competition is that individuals that are close to each other have a higher probability of mortality. And we can imagine that mortality happening across the population. And what's going to happen? Well, we're going to end up again with a uniform distribution, because all neighbors that are close to each other are at risk of being outcompeted by their neighbors. Final example, what about species with wind pollination? Well, for wind pollination, individuals of the same species need to occur close to each other, because that's where wind pollination is most effective. So we would expect a clump distribution for wind pollinated species, and indeed this is something that one observes in the field. These are just some variance to mean ratios for some of the common species at Magaliesberg. You see, interestingly, the alien shrub Lantana camara has the highest variance to mean ratio, suggesting it is the most clumped distribution. On the opposite end, we've got our 2 succulents, Euphorbia shinzii and Allograde hedii, and the common forb Commelina africana. So if you, for your project, you want to quantify interspecific aggregation and see if that can be explained by species life history, then that's one option you could follow. To do this in Excel, you would simply calculate the variance to mean ratio using the var command and the average command. In R, you'd have to take the standard deviation of the data and then square that. That will then give you the variance, and you can divide it by the mean for that same species abundances. Okay, the third question we wanted to test is dealing with interspecific spatial association. In other words, what is the pattern of co-occurrence for 2 different species? Now, we have got 3 broad options here. First, species could be spatially associated. In other words, they tend to occur. We could infer from this pattern several different processes. So firstly, those 2 species that co-occur and are spatially associated might have positive interactions. In other words, the one species creates conditions that favor the other. Spatial association could also result from where species share environmental tolerances. So we'd expect wetland plants to always occur together simply because they require wetland conditions. Finally, we can see species co-occurrence when we're looking at a parasite host or herbivore host plant interaction. And this would occur when species are limited to one host or one food type. And then they are automatically going to be spatially associated. Spatial dissociation occurs when species tend to not co-occur, in other words, they occur in different places. And this could represent species having negative interactions, for example, strong competition, or it could also represent species having different environmental tolerances. So we wouldn't, for example, expect to see succulents and wetland plants co-occurring. So there we would see spatial dissociation. We might also see a random pattern where co-occurrence of species is just as likely as them not co-occurring. And this suggests either that species have little effect on each other, or at least that the net effect, where the positive and negative interactions are balanced out, is close to zero. Now, one way to look at interspecific associations is to examine the co-occurrence patterns of species. So if we're looking at two species, we've got four combinations that we could expect. So if the first is that species A is present and species B is absent, what would be the other three combinations we could have? Well, we could see the inverse where species A is absent and species B is present. Or we could imagine where both species are present or both species are absent. And if we add up the number of sites where one species is present and the other is absent, we can analyze that data using something called checkerboards. Now, it's called a checkerboard because if you have a matrix showing where species are present and where they're absent relative to the absence or presence of another species, you'll see that we get this checkerboard pattern where there are zeros in the top right, top left, bottom right, and ones in the other cells. So it ends up looking like a chessboard. It's worth just pointing out again that we would put a 1 here where one species is present and the other is absent. So those first two rows provide the data that matters when we want to assess interspecific association using checkerboards. So let's just, to help you guys make sure you understand this, let's have a look at an example here. So we've got two species, species A and species B, and they both have 50 occurrences, so they've each been recorded in 50 grid cells. 50 out of the 200 sampled quadrats. So let's look at top left first. And imagine we've got this table where we're showing how species A and B was present and absent. And this is going to show us a strong spatial association. How would you divide the 50 occurrences for each species across that design, across that grid? Okay, and if we see perfect spatial association, all 50 occurrences of species A and B will occur together, and there'll be no cells where one species is present, the other is absent, and the remaining 150 cells will have both species absent. Let's now move down to negative association. What would we expect to see if the two species were perfectly negatively associated? Well, we'd expect that there would be 50 cases where species A is present and species B is present, and likely the inverse also. That then leaves 100 cells where both species are absent. Under random association, the important thing here is that we'd expect to see species A would co-occur with species B, roughly the same time as when it's absent from areas with species B. So again, this allows us to infer ecological process from pattern. When we've got many checkerboards, we say we have a large C-score, and this tells us that there are many sites where one species is present and the other is absent. And this suggests negative interspecific association, although as we've said earlier, this could also, for example, reflect shared habitat tolerances and requirements. So if we asked to ecologically interpret a large C-score, that's what we would do. We'd say, okay, a large C-score indicates that one species is frequently present where the other is absent. And this could be due to shared tolerances, biotic interactions, or even dispersal patterns. Okay, so our final objective is to think about how would we analyze our data to test if there are communities present and if we can find what drives variation in species composition. Now, if we want to identify communities, we essentially need to use classification. Classification in this sense is where we can distinguish repeated groups of species. In other words, we can see what combination of species keeps reoccurring. Second option here, to identify the environmental drivers of species composition, we need to apply an ordination. This is essentially where you look at environmental gradients and you say, what is the order of species and or samples along those gradients? So let's first look at how we would arrange our data to do something like this. Essentially, what one requires is a species by site matrix. Okay, so here we can see every species, its data is only presented in one column, and all the data from one site is only presented in one row. You can see, for example, how at site 4, there's only one species present, and that's species 1. Conversely, at site 2, we've got 2 species present. Species 1 is scarce, and species 3 is abundant. One of the things that you can see about this data set is that it contains a lot of zeros. That's why we call it a sparse data set. And this is simply because many species only occur at a few sites. Now, each species by site matrix can be matched with an environmental by site matrix. So on the left, we're seeing there our species by site matrix. And on the right, you can see there's a matrix. And instead of showing the abundance of a species, it's showing the value for an environmental variable. So let's take a step back and use an example here. If we wanted to predict the abundance of, or the probability of occurrence of a grass based on soil depth, we could plot the data and we could fit a regression line to it, and we could see, okay, as soil depth gets deeper, the probability of occurrence of this grass declines. So that's a really simple model. We've got one response or dependent variable, and we've got one predictor or independent variable. Because there's just one response and one predictor, we'd say it's a univariate model. Now, univariate models are really easy to visualize and really easy to interpret, but they're limited to examining one species at a time. And that's not much use when we want to look at community composition, where there could be hundreds of species that are all our response variables. A model where we've got many predictors and or response variables we'd call a multivariate model. And when we're analyzing our communities, this is important because we want to consider all of our species simultaneously. So just an example for multivariate model, if we were saying that we wanted to understand the occurrence of Acacia caffra, Allograte hedii, Aristida unciformis, and Bidens pilosa as a function of soil depth, grass cover, topography, and aspect, you can see there we've got 4 response variables and 4 predictors. So this is a true multivariate model. Okay, so these multivariate models we could say that they fall under multivariate techniques, and we can identify that this could either be for ordination or it could be for classification. And even there are methods based on dividing data versus methods based on clumping data. Now, one of the important points here is that when we're classifying vegetation, we're assuming there are discrete groups, discrete sets or categories of species that are found repeatedly. In contrast, in ordination, we're assuming that there is a change in species composition along gradients. Okay, so let's start with classification, and particularly with classification, or so-called divisive clustering. And here essentially you start with all your data in one big pool, and then you divide your data into two groups, and then those two groups into divided again. The idea being that this continues until you've got groups of species which are strongly clustered together. Now, typically this has been done in the past using twin span. That stands for two-way indicator species analysis. A two-way just means that you can rearrange your data by site and by species. And that's important for helping us see groups that belong together. Now, divisive clustering aims to identify groups of species that co-occur. Indeed, it can find indicator species that indicate specific groups. So let's take an example. Here we've got a big species by site matrix, and it's difficult to see here where you would divide species or samples logically. And this is where twin span is really useful, because it can help you rearrange your data. And if we rearrange our data, change from species name just to species number, and on the top just to site number, here you can see we can, after rearranging these columns and rows a bit, we can identify that there are two main groups here, the top left and the bottom right. We can then go further and split those again, and again and again, until we reach a stage where there are clear groupings of species. And you can see those groupings of the species on the far right in that dendrogram. Now, in contrast to divisive clustering, agglomerative clustering starts with all of the samples being by themselves, and it then groups samples based on species composition. So the more similar two sites are, the sooner they will be clustered together. So let's try work through this as an example. Here we've got a species by site matrix. And I want you to think here which two sites have the most similar composition, and which site sticks out as different in terms of composition from all the other sites. So what we can see here is, of course, that site 5 and 6 are very similar. They both only have species 1 and only have species 5 together. We also see that site 4 is very different from the others. It lacks species 5, which the other sites have, and it has species 2, which none of the other sites have. And if we were to go through this clustering, this is the pattern we would see. And you'd see that 5 and 6 are the first two that are clustered together, because they have the greatest similarity. And you'll see that site 4 is the last site that gets grouped with these, because it's the most different. Now, what one can do for a dendrogram like this is you can choose an arbitrary threshold and say that everything that is clustered below that threshold forms one vegetation type. So here you could see, for example, that site 1, 5 and 6 clustered together and site 2 and 3 clustered together. It's worth remembering that the threshold value used is relatively subjective, although one generally finds that it doesn't make too much of a difference if you change it a little bit. But that's just something to bear in mind. Now, both forms of classification or clustering assume an underlying, or assume several underlying environmental gradients. Even though these environmental factors aren't used in the analysis, we can use them after the analysis to try and explain why there are the observed differences. So for example, here we might find that site 2 and 3 cluster together based on species composition, and that's driven by them being samples on the south slope. Okay, so that's enough about classification. Let's end by touching on ordination. Now, ordination, as I said, was looking at the ordering of species along a gradient. You could equally say that as trying to understand variation along one environmental gradient. Now, the example I give here is a classic one from a European plant community, and you'll see on the x-axis we go from 1 to 19, and you see that represents going from the bottom of a slope to the top of a slope. You see, for example, like Millennia, here in red, it declines in its cover and abundance with increasing altitude. So it's a species that's limited to the foothills. In contrast, we've got Agrostis canina, which has its higher cover abundance at mid-altitudes. Finally, we could have something like the Vaccinia myrtilis, which occurs across the gradient, but something else seems to be driving variation in its cover abundance. Okay, so it's just an example of how ordination along one gradient would work. Okay, so in ordination, we don't just look at the variation in species along one gradient. We look at how species respond to several gradients at the same time. The trick here is that it's very difficult to visualize multiple dimensions at the same time. In the block on the right there, you can see how the influence of aspect, soil depth, and elevation is considered in a 3D plot. But what happens when you move on to a fourth environmental gradient, or a fifth? And this is where ordination is useful. It allows us to present data in a low-dimensional space. What do we mean by that? Well, it means that even though we can't visualize 4 dimensions, ordination can use information from 4 or more dimensions to summarize what's happening within a community. So during ordination, we really focus on multiple environmental gradients, and we look at how many species respond to those gradients. How does this work? Well, technically we'd say ordination reduces the dimensionality of the data. In other words, it summarizes the main differences between samples into a limited number of axes. And I think the best analogy that I've seen for this is, imagine you've got a very complex network there in the middle of your slide. But you have to reduce it to just 2 dimensions so that it can go into a journal article. So what you could do is you could simply shine a light onto that complex network structure, and the shadow that comes out on the other end provides a 2-dimensional summary of that very complex multi-dimensional arrangement. So this is obviously not a perfect method. Some information is lost, but it's still a useful method. Okay, so let's look at how to interpret an ordination diagram. So the first point here is that the axes are unitless and they're relative. You could flip those axes around, it doesn't really matter. The second point is that the distance between points reflects their similarity in terms of community composition. So species that are located close to each other have similar species composition, whereas when species are separated by plots are separated by a large different distance, it means that they've got dissimilar composition. So here you can see quadrats 14, 15, 16 and 20 clustered together, indicating that they have similar species composition. Likewise, those 6 plots clustered together, suggesting similar species composition, and those 3 clustered together. Okay, so we spoke about environmental drivers. We can also include environmental data on our ordination plots. For categorical variables, we'll do this by just putting down a symbol for the average for the different levels within your factor. And for continuous variables, we can add arrows. So in this example, we can see that quadrats 2, 9, and 12 cluster together. We can also see that there's and the reason they cluster is because of similar composition. We see that the soil moisture arrow is pointing in the direction of this cluster. And essentially what that means is that quadrats to the right hand side are going to have higher soil moisture than quadrats to the left. We also see that the slope arrow is pointing in approximately the opposite direction from the quadrats, suggesting again that these quadrats are located on fairly shallow soils, not steep slopes. So let's look at a far more complex example. This is data from China, where they're looking at 4 different successional stages: shrubs with open triangles going into coniferous forest, mixed forest, and then mature evergreen broadleaf forest. And what we see is that those symbols form a gradient from bottom right, where they're mostly empty triangles, right the way through to top left, where all of the quadrats are these filled cells. We also see that successional stage is indicated as a predictor variable. Interestingly, we see that soil depth is varying in that same direction. Okay, so let's look at an ordination based on data from the Histogram. Here, the color of the symbol represents the aspect of the quadrant that was sampled, and the symbol shape represents the topography that was sampled. And what's quite clear here is that the green points tend to cluster together and the red points tend to cluster together. Not perfectly, but there's a strong trend. How do we interpret this? Well, we could show this suggests that there is a difference in species composition between the north and the southern aspects. It also suggests that because the circles and the triangles and the diamonds don't cluster together, the topography doesn't have a big effect on species composition in the Histogram. Now, we can also add environmental data here. What we see, for example, is that the grass canopy cover arrow points in the direction of most of the green symbols, suggesting that there's a positive relationship between grass canopy cover and the southern aspect, or to put that in more ecological terms, that grass canopy cover tends to be higher on the south slope. The inverse is of course true for the tree shrub cover. We can test this statistically also. For example, see that grass canopy cover indeed is significantly better than chance at explaining variation in species composition. So how would you run an analysis like this if you wanted to do it for your project? Well, I couldn't find any tools in Excel that could do this. In R, however, in the vegan library, and vegan here stands for vegetation analysis, one can find several options, so it can work in R easily. Good, so that's it for the analysis of the data. I now encourage you all to get busy with this project. If you aren't already, start thinking about what your research question is, how you would analyze it, and how you'd end up discussing it.